Good morning. Today we're going to discuss chapter 4, which is the next step after talking about cells. We're going to talk about tissues. There are 10, 20, 30 different types of tissues in the human body, and we're going to touch on probably 15 or so. The most prevalent or the most popular? No, prevalent, I guess is a good word. So again, why this matters is a video that you can open up and view either here or by accessing Mastering. And what we want to do is talk about tissues and sort of discuss why we need to know them, right? What do we need to know about the tissue that gives us an idea about the function of the organ, etc. So when we talk about tissues were called tissues, the living fabric, like how the organs are put together, how the bodies are put together. And we've already talked about individual body cells, right? A red blood cell is different from a neuron, is different from a bone cell. And now we're going to talk about tissues. So muscle tissue is different than cartilage, is different than skin tissue, right? And so we're going to, discuss those differences in the structure and the function. Going back to chapter 1 with the complementarity of structure and function. We're going to look at the structure of a certain tissue and see how that dictates what that tissue is able to do. In lab, we're going to look at the histology. We're going to study tissues on slides. And somewhere in the middle of this chapter, there's a little bit of a discussion on histology and how slides are prepared. So when we divide the body up into tissue types, we're going to talk about the 4 basic tissue types. In this image, it starts with nervous tissue, the brain, the spinal cord, the nerves, about internal communication. We then have muscle tissue, which is about movement. We either have skeletal muscles, which move our bones, cardiac muscle, which contracts the heart, and smooth muscle that contracts the walls of hollow organs, like our digestive system and our bladder. Epithelial tissues forms boundaries between certain environments. And also protects, secretes, absorbs, filters. The epithelial group is pretty varied. Then we have our connective tissues, supportive. So our bones are connective tissues. Our cartilage is a connective tissue. Fat is a connective tissue. Support, protect, binding things together. So we're going to sort of tease out the general characteristics of the group, and then we'll talk about individual tissue types. So you'll need to know this for both the lecture and the lab. So when we look at tissue in the lab, what we're going to look at are stained slides. And the tissue has been fixed, sectioned, and stained. So some sort of human who has deceased, some of their tissue has been fixed, meaning it's preserved, that it's been sectioned into teeny, teeny, tiny slices, laid on the slide, stained. Some of the slides we're going to look at will be blue, some will be pink, some will be kind of a brownish, some are purple, depending on what we want to see. Different dyes are going to highlight different structures. So we'll look at all different kinds of things. So fixed, sectioned, and stained, and then we can take a look at them. The one thing we have to remember when we're looking at tissues under a microscope is that tissues don't exist in a vacuum. So for example, if I was going to take a sample of the tissue from my thumb, I might get some epithelial tissue, might get some fat. If I went deep enough, I might get some muscle. Depends on how I cut it. Am I just taking a sample from the surface, or am I going to cut down and take a cross section through this whole area? I would get skin, fat, muscle, bone, right? So I get all kinds of tissues. So even though the tissue slide says it's simple columnar epithelia, there's probably 2, 3, 4, 5, 6 different tissues on each slide, but we'll talk about that in lab. So we're going to start out with epithelial tissue as our first grouping. I think we're going to look at, let me check my notes here, 6, 7, if we talk about glandular epithelia. So we're going to look at 7 different epithelial types. They come in 2 groups, one covering and lining. We're going to cover something, we're going to line a cavity, we're going to create that boundary. And then there's glandular epithelia, epithelia cells that secrete, salivary glands, your thyroid gland, things like that. Notice how it says the main functions are 6 things, 6 main functions: protection, absorption, filtration, excretion, secretion, and sensory reception. Now, what you need to remember is that not all epithelial tissues do all of the things. Skin, really good at protecting, right? Providing that barrier between the dry air on the outside and the moist on the inside of the skin. Very good protector. Is it good at absorbing? No, right? It's a really good protective barrier, right? We have the delicate lung tissue, very good at filtering, right? And absorbing that oxygen, carbon dioxide. But is it good at protection? No, very delicate. So just because those are the 6 main functions of the group doesn't mean that each individual tissue type doesn't have its own specific functions that it does, very well. So when we talk about epithelial tissue, there are certain characteristics that all epithelial tissues have. And one of those things is called polarity. And when we talk about polarity in epithelial tissue, we talk about the fact that there is a top and a bottom. The top would be called the apical surface. So you can touch the apical surface of your stratified squamous epithelia on your skin. Apical, upper free side exposed to a surface or a cavity. The lining of your mouth is also stratified squamous, right? But it is an apical. It's exposed to a cavity. The basal surface is the attached surface. So this epithelial tissue is attached to the tissue underneath it, all right? And it touches that next connective tissue on the basal surface. So we have a top and a bottom, or an exposed side and an attachment side. This is a picture of some simple squamous epithelia. You'll notice those tall rectangular cells with the purple nuclei all in a row. This is the apical surface that borders the open space, and the basal surface that's connected onto that sort of wavy looking connective tissue. What else is characteristic of epithelial tissue? They have what are called specialized contacts. If we go back to this picture, notice how each of those cells is lined up like my fingers. Tight, 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 tight. Notice the tissue underneath that. There's some cells in there, but there's a lot of space. Epithelia, tight, tight, tight. Specialized contacts. Many form sheets. We're making a barrier, remember. This isn't a barrier. This is a barrier, right? So they are held together by what are called tight junctions and desmosomes. Now, epithelial tissue doesn't have a lot of support. So underneath every single epithelial tissue layer is some supportive connective tissue. So here you can see that border of simple squamous epithelia, and underneath would be the supportive connective tissue. The epithelial tissue is avascular. When we say avascular, it means without vessels, without blood. So our skin, the top layer, the epithelial layer, doesn't have blood vessels. I can scratch like this, scratch, 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 no blood, right? But there's a lot of nerve fibers, which is why we can feel a little bug touch our skin, or if we brush a hair across our skin, we can feel that, right? So we do have a lot of nerves. So no blood vessels, but nerves. Very high regenerative capacity, right? If I cut myself within a couple days, this would be all healed up, a little scab for a bit, and then it would heal itself up. Very, very, very high regeneration rate. All epithelial tissues, or most epithelial tissues, have 2 names. The first name is how many layers there are, how many cell layers there are. So when we look at this picture, there's 1 cell layer. Tall, rectangular cells all look like this, 1 layer thick. That would be a simple epithelia, 1 layer. Stratified epithelia is going to see layer upon layer upon layer. That's called stratified. The one that we looked at was simple because it's 1, and columnar, very tall column-like cells, right? This is a shape, so it's going to be simple squamous, simple cuboidal, simple columnar, or stratified squamous, stratified cuboidal, etc. So we're going to have number of cell layers and the shape. This is a picture of simple squamous on the top, simple meaning 1 layer, squamous meaning flat. On the bottom, stratified squamous, many, many, many layers, flat cells. These are the shapes. Squamous is flat, cuboidal is like a little box, and columnar is like a tall column. Now, what you're going to notice when we're looking at these, if we go back to this slide again, some of them are bent, some of them are kind of not exactly column shaped, right? We're a very soft tissue. We don't have a lot of tissues that are solid. So even though it says cuboidal, don't look for actual boxes like a plant. We're looking for something that's kind of box-like. These are something which are kind of column-like, right? So when we look at these, don't expect them to all look exactly like that, but there's sort of a shape classification. So now we're going to go over each of the epithelia specifically that you need to know, the characteristics, the function, and the location. So this one is simple squamous, one layer, flat cells. It makes it logically looks like it would be easy for things to pass across that. So if you'll notice the function, it says diffusion, filtration, in a site where protection is not important. That little skinny flat layer of simple squamous cells is not protective at all, very delicate. The picture on the right shows the air sacs of the lung tissue. Now, when you're looking at that, you'll see these wavy lines. Those are simple squamous cells attached to each other around a space of air. So that light purple background is air, and the cells are in a line around. So again, think of a balloon. The simple squamous cells would be the balloon, the air would be in the middle. These are the cells, all right? You're going to find them in the kidneys, the lungs, the lining of the heart, all right, and the membrane called serosa, which we'll get to in a bit. Simple cuboidal. Again, we're looking for one layer, cube-like cells, still thin, so they're very good at secreting and absorbing. We're going to find them in our kidney tubules, some of our glands, and on the surface of the ovary. When you look at the image to the right, it looks like there's a bunch of big round things with dots in them. Those round things are tubules. And so picture it being like a mess like this of all different tubes, and my fingers are all different tubules. And what you've done is you've cut it, and now you're looking at the ends. And so now you're looking at the cut ends of tubes. And in that circle are all simple cuboidal cells around it. So the one that it's pointing to, right, it looks like kind of a squished tube. You can see 5 or 6 different cells in there. Right, so those are our simple cuboidal cells. Here we have our simple columnar like we saw before, 2 layers of it. And the white stripe in the middle is going to be the lumen, or the opening to the cavity of the intestines. These are still good for absorption and secretion, not protection, right? And these are going to be tall column-shaped cells. We're going to find them in a lot of different places. Digestive tract, gallbladder, some of our glands, our small bronchi, uterine tubes, and subregions of the uterus. Notice a couple of new words here, ciliary and non-ciliated. Cilia is going to be these wavy extensions of cytoplasm that move stuff along. So the ciliated type under the function paragraph says that it propels mucus. Now, if you're going to find it in the small bronchi in your lungs, it's going to push the mucus up out of your lungs, right? That's what the cilia is for. Now, when you think about the non-ciliated type, which you have a picture here, that means just cells that are like this, no cilia on the top. We don't want to move the food by. This is the intestine. We want the food to sit right here and get absorbed into the large intestine. So you wouldn't want cilia in the intestine. You want cilia in the lungs to get all the goo and mucus out of your lungs, but you don't want it here. So cilia only where it's functionally important. Then we have this crazy looking thing. This is called pseudostratified columnar. Pseudostratified. It looks as though, when you look at it, even in the yellow cartoon and in the pinkish red picture, it looks kind of crazy. It looks as though there's columnars stacked up all over it, but actually these are one layer of cells with differing heights. They all start at the bottom, and they all, most of them will reach the surface. Some will not. But there's not stacked on top of each other. Interesting little tissue there. You'll notice some pretty prominent cilia on the top of that. Look at those fuzzies, right? And notice where we're going to find them. The ciliated variety lines the trachea, again, getting that mucus up, and in the sperm-carrying ducts, right? We're going to move the sperm along, and in some of our large glands. All right, then we start moving away from the absorbing and secreting portion of the program. We get to stratified squamous. The entirety of the pink and red that you're looking at in that slide is the many layers of the stratified squamous epithelia. This is about protection from abrasion. This is stratified squamous. So I can do this all day long, right? And my cells are flying off, but there's layer upon layer upon layer, right, that I have as a protective abrasive mechanism. So this is going to be the keratinized, right? This is the outer dry epithelia. keratinized stratified squamous. Inside the mouth, esophagus, where we're going to have a lot of food and teeth and tongue scratching. This is stratified squamous, non-keratinized, right? A moist membrane, but still protective. Transitional epithelia resembles stratified squamous and cuboidal. It kind of looks lumpy when it's flaccid or not full. But when it stretches, they get really, really, really flat. Transitional transitions from cuboidal. This is found in the bladder and the ureters. So when the bladder is empty, they look cuboidal. When the bladder is full and stretched out, they stretch to become flat. It's transitional epithelia. Glandular epithelia is one or more cells that make or secrete a fluid, any kind of a secretion. So we could talk about hormones, sweat, oil, saliva, all kinds of things. Anything that our glands will secrete all over the body, right? Salivary glands in the mouth, sweat glands in the skin, goblet cells that secrete mucus pretty much in our entirety of our digestive tract. Let's see. Hormones, right? Our thyroid or adrenal glands, all going to be glandular epithelia. We have different types of classifications of glands. You can have a unicellular gland or a multicellular gland. So one, like a goblet cell, is its own gland as one cell, and a salivary gland is a grouping, a multicellular gland that will secrete, for example, saliva, adrenal, etc. They can release their secretions endocrine, internally, like hormones, or exocrine, like sweat off the skin, exit, outside the body. Now, one of the things that can be confusing is some of our exocrine glands, like the pancreas, for example, is both endocrine and exocrine, depending on what we're talking about. The exocrine function of the pancreas secretes digestive fluid into the small intestine. Now, you might think that's internally secreting, which it is inside the body, but technically the digestive tract from mouth to anus is one continuous tube open to the outside. So that's considered external. The pancreas also secretes insulin and glucagon and other hormones in an endocrine function into the bloodstream. So exocrine can be a little confusing because one would think that the intestines are internal, but from a glandular standpoint, they're not. So this is a multicellular exocrine and endocrine glands. You can see how there is an exocrine gland cell right in the middle. The epithelial glands, which are on the left, right? They just go down, and we have this little sort of glandular, like a little tunnel. The exocrine glands itself kind of have a little gland at the bottom, like a little cavity. Then we have the endocrine glands. Notice that on the right-hand side that have blood vessels running through them so the secretions can go right into the blood. And we will sort of further define endocrine glands. Endocrine glands are going to be ductless. They're going to secrete by exocytosis. So we just talked about in chapter 3. We're going to create the secretion, and we're going to push that vesicle to the membrane and then expose that outside the cell by using exocytosis. Endocrine glands are going to be secreted onto the body surface or into a body cavity. We have way more exocrine glands than we do endocrine, but they're usually really tiny. Mucus, sweat, oil, salivary glands are all examples of our exocrine glands. We can have uni or multicellular. So there's some, differences between the different types. So this is a unicellular exocrine gland called the goblet cell. All right, the goblet cell looks like you could hold it from the purple, and it looks like a goblet. And it is going to produce what's called mucin. And mucin is a sort of precursor to mucus. Mucus is in our intestinal tract, in our respiratory tract, right? The point of mucus is to protect. In the stomach, for example, the mucus protects the stomach lining from the acid environment of the stomach. In the lungs, mucus catches anything that you breathe in, right? You go, in that breath, I just breathed in skin cells. I probably breathed in some dust, some bacteria, some viruses, some fungi, who knows? But the mucus that lines my mouth and my respiratory tract should capture those things. And then the cilia is going to bring it all up so I can go and swallow it down. So goblet cells, pretty important little guys, but they're unicellular exocrine glands. So then we also have multicellular exocrine glands that vary based on structure and their mode of secretion. Here we have simple ducts, compound ducts, and then we have the alveolar. So ducts are going to be more straight, alveolar are going to be more rounded. That's pretty much the difference. Right? You'll notice how they all open onto surface epithelia. They can be simple, simple branched, or compound, right? Either way. The one on the bottom right actually shows a tubulo-alveolar, right? The different shapes. So it's basically just about how many branches, if any, and then what the shape is. But again, multicellular exocrine, many cells secreting onto the body surface or into a body cavity. So how do these multicellular exocrine glands secrete? We have what's called merocrine, holocrine, and apocrine, different types of glands. Exocytosis are merocrine. If we're going to rupture the cell and spill the contents out, that's called holocrine. And then apocrine, we're going to accumulate the products, but we only sort of rupture the top, or the apex actually, that'd be the bottom. We only rupture the top and then it heals back up again. Now, some people think that maybe some of our mammary glands, our breast milk cells, are apocrine. Jury's still out on that, right? Not really sure, but merocrine and holocrine, very, very prevalent in the body. This is a picture of a merocrine gland, right? So secretory vesicles, and then the secretion goes up the duct and out, right? So if we look at merocrine, products secreted by exocytosis. So you'll notice all of the little yellow secretory vesicles gathering on that surface. They're going to spill their contents into the gland and then up and out. The one on the right, we have all of the cells stacked on top of each other, and actually the cell itself ruptures open. We're still secreting something, right? Just depends on the type of gland and what the secretion is. In the holocrine gland, that classic example is our our oil glands, right? We get on our face, right? Our T-zone, our sebaceous oil glands. Right, so merocrine and holocrine. I'm exhausted already. We just have only done the epithelial tissue. So if you want to take a break and come back and do connective tissue at another time, this would be the time to hit pause and go do something else for a bit. So connective tissues are found throughout the body, most abundant, widely distributed, binding, supporting, protecting, insulating, storing, reserving fuel, and transporting substances. Again, a lot of different functions. Connective tissue doesn't have a lot in common, right? If we think about just the three that you know the names of that are listed here, we have connective tissue proper, which we're going to go into. They have specific sort of 6 or so connective tissue propers that we'll discuss. Then cartilage, bone, and blood. And when you think about bone, everyone knows what bone is. Calcified, hard, structural, internal support. White, rigid. Blood, liquid, red, has nothing to do with structure, carries oxygen. How is blood and bone both connective tissues? So connective tissues don't have a lot in common. What they do have in common is this. They all arise from the same embryonic tissue called mesenchyme, which is why they're put together in the same grouping. They have varying degrees of vascularity, so that's not really a common, commonality. Cartilage is avascular, just like epithelial tissue. There's no blood supply in cartilage, but bone, tons of blood in the bone. And then cells have what's called an extracellular matrix. There's fiber around them, right? There's something else going on there. So they have a ground substance, fibers, and cells. The ground substance is what it is. So when we're talking about bone, the ground substance is a hard calcified collagen matrix. That's the ground substance. The collagen would be the fibers, right? And the cells are going to be osteocytes inside that bone tissue. When we're talking about blood, blood's ground substance is the liquid plasma. Fibers are the fibrinogens that would help with blood clotting. And the cells are going to be red blood cells, white blood cells, etc. So they have 3 main elements, but totally different functions and totally different how they're put together. I skipped these two comparison tables, and this is good to come back to when you want to get a sort of an overall, right? Table 4.11. You can find this in your book. You can find it on Mastering. And it basically gives you connective tissue proper, cartilage, bone, and blood. Tells you about the cells, the matrix, and the ground substance. So you can come back to this. I'm not going to review it now because we're going to go over each cell type in very much detail. So the ground substance, right? The space between the cells, either a solid or a liquid. The connective tissue fibers are going to either be collagen, elastic fibers, or reticular fibers. Different types of support. Collagen, the strongest, most abundant. Elastic, stretch and recoil. And reticular, very fine, very delicate. And we'll talk about where we're going to use each one. The different types of cells. Each type of tissue is going to have a different type of cells. If you see the word blast, it's an immature cell that's actively secreting and creating. If you see the suffix "cyte," that means it's a mature, less active cell. It's about maintenance. So when we're talking about bones, osteoblasts, osteo is the root word for bone, or the Latin for bone. So osteoblast is going to secrete the calcified matrix to create bone. The osteocyte is going to live within that matrix and maintain the tissue. All right, so two different types of words. We're going to have fibroblasts, chondroblasts, osteoblasts, and then the cyte versions of each one of those words. Other types of cells that we might find are going to be fat cells, white blood cells, mast cells, macrophages, and we'll go through each tissue type and tell you what's found where. This is a generalized picture of connective tissue, showing the fibers, the ground substance, and cells. Not all of these cells are going to be in one tissue type. Fat, mast, neutrophil, fibroblast, macrophage, and lymphocyte. No, this is a generalized idea. So let's get into the specifics. When we talk about connective tissue, this is a nice sort of a stepwise way to group them. Connective tissue can either be connective tissue proper, in which we will go to loose or dense, and then 3 each. Or it can be cartilage, of which there are 3 types, bone, and blood. So we're going to talk about all 11 of these, from areolar, adipose, reticular, regular, irregular, elastic, hyaline, elastic, fibrocartilage, bone, and blood. Notice how elastic is on there twice. There's elastic cartilage, and there's dense elastic connective tissue proper. Don't get them confused. All right, so connective tissue proper is all the connective tissues except for the other ones that are listed. Loose connective tissue means it has a lot of space between fibers, which we'll see. Dense means those fibers are really tight together. So this is what's called areolar connective tissue. You'll notice how there's a lot of fibers, all the black and sort of pinkish lines, elastic fibers, collagen fibers. There are some cells. So notice how this is very different from the epithelial tissue, because in this, the only cells are the black dots. Remember when we saw epithelial tissue, all the cells were jammed up against each other. So this is areolar connective tissue. It looks very woven, right? Lots of fibers. We're going to find it under the epithelia of the body. Well, that's everywhere, right? Wherever we have epithelia, we're going to find areolar connective tissue. As part of the lamina propria of mucous membranes, surrounds capillaries, packages organs, so you can pretty much be sure you're going to see areolar everywhere in the body. This is an interesting looking guy. These are fat cells. Each one of the big round circles is a fat cell. Some of them you can see the nuclei, some of them you can't. Same matrix as areolar, right? But very sparse. Most of these are going to be big fat cells crammed together. And then again, this is fat, so stored energy, insulation, cushioning, protecting the organs. We're going to find it under the skin, within the abdomen, right? All over the place. So it's loose connective tissue, adipose. This is also a loose connective tissue. It's called reticular. The black looking tree branch looking things are the reticular fibers. These are very delicate though, so it's going to support the lymph nodes, the bone marrow, and the spleen. Going to support those red blood cells. You can see all kinds of cells on the in the picture there, white blood cells, and a lot of space. A lot of space. Now notice this one's different. We've now gone into the dense connective tissues. Notice all that pink is all fibers. And a couple of the purple lines are going to be the fibroblasts. Not a lot of cells, not a lot of space, awful lot of collagen. Collagen, very strong. When we think about dense regular, notice how the fibers are moving all in one direction. That's very, very good tensile strength in one direction. Right? So there's going to be tendons, ligaments, things like that. A lot of pressure. Then we end up with this guy. Notice how it's all wavy. The blue in this case is the collagen fibers. Here we have red collagen fibers. Here we have blue. But notice how they kind of go left to right, up and down. They go in like this. This is tension in a lot of different directions, right? A little bit different of a function. All right. It's going to be fibrous capsules around the organs and the joints where we want to be able to have movement in different directions, but structural support. This crazy looking thing is elastic, dense elastic tissue. Again, these fibers are very tight together, all in one direction, a little bit wavier. Right here we are allowing for stretch and recoil, but support. So the arteries, the ligaments, right? We're going to have the arteries of the large arteries that stretch with our heartbeat, right? We need support. So the elastic connective tissue helps with that. So we're moving out of the connective tissue, dense and loose. Now we're into cartilage. Now cartilage is interesting because it has an amorphous but firm matrix, right? It's not bone, but it's got some play to it, but it's definitely a firm, solid matrix. Each one of the black dots that you see sort of surrounded by that white halo is a chondrocyte. So chondro means cartilage. This is a mature cartilage cell. Inside what's called a lacuna, it has like its own little cavity there. So where are we going to have cartilage? This is hyaline cartilage. So hyaline cartilage is going to be at the ends of all of our long bones. It's going to be at the costal cartilages of our ribs. It's going to be in between anywhere where a bone meets a bone. All right, little cartilage of the nose. All right, hyalins all over the place. This is elastic cartilage, right? We have two spots, the external ear. So if you take your ear, which I know you all just did, and you bend it around, right? It has some structure, but you can always bend it, right? It's much more flexible than the cartilage of your nose. All right, so this is elastic cartilage. Again, you're going to see the chondrocytes, you see the lacuna, you see the matrix, right? This is a little bit more flexible. Then we have this guy. This is fibrocartilage. Fibrocartilage is going to be the intervertebral discs between your vertebrae and your spine. It's going to be at the pubic symphysis, where two halves of your hip bones come together, and in between the femur and the tibia on the in your knee. These are tensile strength and a compressive shock. Anywhere where there's going to be a lot of pressure like this. All right, so a lot of fibers. Chondrocytes again, right? So this is fibrocartilage. Then we move to bone. Bone's an interesting one from a cellular perspective. It's very easy to see that it's bone. It looks like rings on a tree. Each one of those sort of units of circles, right, is called an osteon. In the middle, there's three on this slide. That's not a nucleus. That's the central canal. That's an artery, a vein, and a nerve. Each one of those black lines in the circles is an osteocyte. That's the cell. Bone is bone. We know where it is. Then we have blood. Blood has a liquid matrix. You can't really see the fibers because they are dissolved. You see red blood cells there. You see some white blood cells, right? And blood's role is to carry oxygen, nutrients, waste, etc., as well as move the immune cells around the body. All right, third type of tissue. Again, if you want to take a little bit of a break, we're going to do muscle and nervous tissue, and then tissue repair as our last segment of this PowerPoint. So muscle. all about movement, highly vascular, skeletal, cardiac, and smooth. Easy. Skeletal muscle, if you're looking at the picture, the muscle cells are the long stripes going left to right. Top to bottom, you see the stripes, those are called striations. The interesting thing about a skeletal muscle cell, notice what it says there under the description, long, cylindrical, and multinucleate. It's one of the cells that we have that has more than one nucleus, which is interesting. Skeletal muscle attaches and moves our bones. So my bicep, my tricep, my brachioradialis, right, are my flexors and extensors. These are all skeletal muscles, voluntarily controlled movement of the body. Cardiac muscle looks kind of similar. You'll notice how the cells go left to right. You'll notice there are some striations, but the main difference for cardiac is that they're branched, and you can see that word, intercalated discs, those solid looking lines that you see. And you'll see how the cell kind of branches, and so what we want to do when we have a heart is contract as a unit. Here, I want to pull this to this, my forearm to my shoulder. All I'm doing is contracting one direction, right? But the heart, we want to squeeze, squeeze, squeeze. So what that means is that these cells have to branch around the heart. So we all squeeze together, right? So they're not all in this direction or not all in this direction. They go like this and the whole heart can contract, right? So that's cardiac muscle. Smooth muscle, totally different, right? We don't have those striations, uninucleate cells. They're all tapered, skinny, right? What we call sort of an almond shape, spindle shaped, right? Totally different. Here we're going to propel substances through a tube, digestive tract, bladder, something like that. Walls of hollow organs. And then there's nervous tissue. For nervous tissue, we have the two basic groups of cells, the neurons that conduct the impulse and the supporting cells that insulate, support, and protect the neurons. Neurons have evolved away from having a lot of protective mechanisms. They're very much just some plasma membrane with a nucleus. That's kind of it. So they need much more support, insulation, and protection because they've evolved to be very specified for impulses. So here we have the big orange kind of globby things are the cell bodies of these neuron cells. Then all of those little strings are neuron processes. So unlike our, simple cuboidal is just a cell like this, or a muscle tissue, long cylinder, these guys have sort of a head end like an octopus, and then neuron processes. Right, so it's a little bit of a different type of cell. Notice in this picture we have some supporting cells, those sort of golden cells that are surrounding the neurons. We're going to find nervous tissue in brain, spinal cord, and nerves. All right, now, when we talk about tissues, sometimes two tissues come together to form not really an organ, but sort of a second level tissue. And this is where we're going to talk about membranes. We have three different types of membranes in the body: cutaneous, mucous, and serous. So cutaneous membrane, our cutaneous membrane is our skin. It's two layers, keratinized stratified squamous epithelia attached to the dermis. Now the dermis in this case is going to be both areolar connective tissue and dense regular connective tissue. It's a dry membrane. Then we have the mucous membrane. So the mucous membrane is any cavity open to the exterior. So that's going to be our digestive tract, our urinary, right, because our bladder is open to the outside, and our respiratory. Any place that's open to the exterior. Oh, and in females, it's also going to be the reproductive tract. We're going to have epithelia overlying what's called the lamina propria. Now, we can't be as specific. Here we said it's keratinized, stratified squamous epithelia, because that's all that it is. When we're here, though, we can't make that classification because each region is different. For example, the mouth is stratified squamous epithelia over the lamina propria, which is more than likely the areolar and dense irregular, for a lot of support. In the delicate lung tissue, we have a epithelial sheet of simple squamous, right, overlying a very delicate lamina propria. So every mucous membrane is going to have two different epithelia and connective tissues together. Then we have the serosa. The serosa are in the closed body cavities. So our pericardial cavity, our pleural cavity, our peritoneal cavity that we talked about in chapter 1 are simple squamous resting on thin areolar. Very, very, very thin. The only job for this is for reducing friction, slippery, and moist. Right? We have pleura, pericardium, and peritoneum. Simple squamous over thin areolar. And the last thing that just chapter 4 talks about is tissue repair. And tissue repair is when the body's barriers are compromised. So we can get a cut on my arm, I can bite the inside of my cheek, I can, get in some sort of an accident and tear, my lung tissue or tear a liver or your spleen or something like that. Tissue repair is tissue repair. The example that we're going to go through now has to do with the skin. However, it's all the same thing: inflammation, organization, and regeneration. Most of the time, we're going to end at regeneration. Sometimes we end up at fibrosis, and fibrosis is when you get a major injury, when the connective tissue replaces the destroyed tissue, you get a scar, and the original function is lost. So we can regenerate. So if I cut this a little bit, and then in a couple of days it scabs, and then it goes away, and you won't even know what happened. If it was a big one, right, I might end up like I have a little scar right here from a dog bite. It's like a little tooth right there. And that was a deep one. So that actually was fibrosis. There's a little scar there. But inflammation is step 1. Inflammation is the release of the inflammatory chemicals. We're bringing fluid. We're bringing immune system cells. We're bringing white blood cells and plasma proteins and all of that to the area. That's inflammation. All right. The next is going to be organization. So we got to figure out how we're going to get these two pieces of tissue to go back together. So we have to organize. We have some new blood-filled tissue that grows. Epithelium begins to regenerate. Collagen bridges the gap. And then at the end, the scab and any of the kind of repair things, scaffolding is pulled away. So this is organization. So you'll notice that we have fibers. It's called granulation tissue coming across. We've got fibroblasts and macrophages, and things are starting to knit back together. That's the regeneration, the organization part. Now that we've reestablished the blood supply, we've closed the wound, we're going to regenerate it. All right, so the regeneration means that there's a scab, there's fibrous tissue, this thickening epithelia, and hopefully when the scab detaches, the skin or the injury is permanently repaired. All right, so this is regeneration and fibrosis. So you'll see there's a fibrosed area underneath, right, that has that sort of white scar looking thing, and on the top looks fully regenerated right from the skin. Sometimes that happens, sometimes that doesn't happen. All right, so but the tissue repair process, inflammation, preparing the area, bringing all the things that needs to be there to fix it. The organization, we're going to start from the bottom, we're going to fill it in, we're going to pull it close together, and then we have the regeneration. We're going to affect that permanent repair, pull all that scaffolding out of there, and now it should look as though nothing ever happened. But this doesn't happen the same way in every tissue. Some tissues regenerate really, really, really quickly. Epithelia, bone, dense irregular, anything like that. Tissues with moderate, right, can sort of fix-ish. Smooth muscle, dense irregular. Some tissues can't repair at all. Heart, nervous tissue, things like that. Now, why is this? When we think about the tissue types as they're listed in this slide, the ones that regenerate extremely well, they have a very, very, very well vascular, nutritionally supportive, right, area. Even though epithelia doesn't have blood in it, it's very well supported by the connective tissue underneath it. Bone, very vascular. Areolar, very vascular. When we think about cardiac muscle, nervous tissue, and brain, no regeneration capacity whatsoever. Why? Right? Cardiac muscle is very vascular, but it doesn't repair itself. So these are things that scientists are working on, right? Regenerating nerves for people who are paraplegic, quadriplegic, etc. Getting, after a heart attack, when that piece of your heart dies, right? They want that piece of heart to sort of come back to life or regenerate. Right now that doesn't happen. So that's sort of a new area of research, or up, sort of, they're always working on getting tissues to regenerate. So why do we care, right, about tissue types or tissue repair, anything like that? If the scar tissue, right, that forms in the heart, well, maybe your heart doesn't work very well. Maybe your heart is not going to be able to expand or contract. So that will really seriously reduce the volume, right, in your of blood that can go through the heart. Scars, right? Scars and adhesions, right? If you tear something or rip something or have some sort of surgery in your abdomen and you get an adhesion, as the tissue is healing, maybe there's a scar. Maybe that scar grabs onto another organ and then it starts to twist, right? So we need to know about tissue repair when we are trying to figure out from a clinical standpoint what might be going on with the patient. Okay, so that is the end of chapter 4. I know it was a long one, and I hope that you enjoyed it, and I will see you for chapter 5.